anybody who was here yesterday, then certainly there'll be some repetition in the beginning. So I think we may as well get started and get this day 2 on the way. Next slide. So that's me. My day job, I'm the CEO of Global Alliance for Genomics and Health, and really from the setup of this, been co-chairing IHCC along with Jeff and Terry Manolio, and bringing the power, the presence of G2MC and GA4JH to backstopping and helping launch IHCC. Next slide. This is a formal welcome to day 2. I'm just going to take you through a meeting introduction and a brief day 1 recap. Next slide. Again, just always worth pausing and repeating the vision, particularly for anyone who wasn't here. I wasn't able to join us on the first session on Monday. And the sense of a global networking, it says large cohorts, and I hope by now everybody knows that large is relative. And if it is in an underrepresented population, that threshold lowers. What we're really trying to build here is something of global value and advance human health through that value. Next slide. So from day 1, some of this was the stuff that any organization has to go through. Might not be the most exciting, might not answer the greatest scientific questions, but it's the necessary, important things you have to go through in order to form a viable, sustainable organization. And we did some great updates and progress on membership agreements, the roadmap to governance implementation, the dry stuff. And then we got into more of the feeling the meat of the work we do and the policy and biodata sharing updates. Laura and others, and that sense of now translating a membership agreement and a governance framework into the actual policies that will shape the way we share data. Talked about mobilizing the IHCC cohorts to address COVID-19, not something we had on our plans when we met last time we met in person, but something that's been extremely important and current in the way that we've been able to pivot quickly to bring efforts from the cohorts to addressing this global pandemic. I think there's a great representation of different things, just really the tip of the iceberg of what cohorts can do towards dealing with whether it's an urgent global health issue or the more chronic ones that we'll get back to, hopefully eventually. And then we went into the first of the strategic planning workshops. So in my group, it was a great size to be at that number of people and with the backgrounds that we had. So it felt like a very useful workshop where everybody got lots of opportunity to give their input and give their feedback. So we're going to go into today's meeting objectives. Obviously the build and the follow on from day 1. Firstly, to revisit the objectives and key actions resulting from our last virtual summit and update IHCC members on progress to date, and to engage the entirety of the membership in developing the key topics on that strategic plan. So very much a continuation of the approach we took on day 1, but with new content. Just a refresher for everybody, these were the objectives as we went into the May 2020 meeting. Today, we're going to focus on those bottom 2, on 3 and 4. And immediately after this introduction, we'll move on to an update on the cohort atlas and the great work that's been done there. And then we'll come back to that all overarching world of engaging the IHC membership in developing the topics for our scientific agenda. Okay, highlights of what we actually came out of the virtual summit with. That were the objectives we went into it. Here's what we came out of it. And I think you see as we're walking our way through that, clearly today, we're going to continue this, or over this meeting, we continue the work we've done on strategic planning. So now we're into, that's the background, that's the recap of where we've been and how we got to today's session. Now we're moving into session 3 on our post-summit progress. And we'll focus, as I said, in this session on summit actions 3 and 4. You can see them there. And the very first of those is to bring back to the fore the work that's being done on the cohort atlas. And you'll see there in terms of this session outline, where are we today? What lessons have we learned from the pilot? And what happens next? How do we expand this from the pilot project into the scale up? I see at least 3 of those 4 individuals. I'm not sure that is Thomas here yet, but anyway, this has been led under Philip and Thomas and been implemented by Christina and Melanie. Here's the objective from ISC3, and you'll see that in terms of that first big, meaty, long one, all kinds of status, but it is in progress and it is ongoing. And that I think is, we've done great work to date, but we have a lot to do in order to actually populate this and make it come to life and make it really useful for everyone. And then we'll get an update on that resource center with relevant data tools and cohorts. Okay, so the cohort atlas, data atlas. As I said, a lot of the heavy lifting done by Christina Young from OICR, and her partnered in the build-up of this with Melanie Courtot from EBI in the UK. And I think they were doing this within our data working group. That working group is led by Phillip Awadalla, also from OICR, and by Thomas Keane from EBI. So with their leadership and the great direct contributions of Christina and Melanie, we've moved forward. And I think it's over to the team. Take it away.
Thanks, Peter, for that great intro and start to this meeting as well. So I'm Phillip Awadalla, as Peter has mentioned. I'm at the Ontario Institute for Cancer Research, and I'm at the University of Toronto in the Molecular Genetics Department. And as Peter had mentioned, I'm co-leading this activity with Team A, with Thomas Keane, who's at the European Bioinformatics Institute. And I'm going to just be doing a really quick introduction here. I don't want to take up too much time here, but the IHCC Data and Infrastructure team is Team A, and we're challenged with the opportunity to create an interoperable platform for the IHCC, capturing data from at various levels from the different cohorts, largely starting with metadata, such that we create, if you like, a visible platform of data and data tools that can be used by the greater or broader research community. So just really quickly, what you're going to be hearing about over the next 30 minutes, Christina is going to walk us through the developments of the IHCC Atlas browser. We heard the first, if you like, instance of it in our May meeting, and so we're going to be getting an update on that activity during her live demo in the next few minutes. Melanie Courtot, also at EBI, is going to be walking us through the harmonization activity, the metadata harmonization activity, updates on that activity, and then we're going to end off this session with a round of a summary along the lines of what we're thinking about with regard to global strategy from Thomas Keane at the end of this session. So, Christina, I'll turn it over back to you.
All right. Thank you, Philip. So I'm going to start with talking about the Atlas browser. Like Philip said, back in May, you saw a demo of the Atlas browser. That was our first stab at it, and it was a prototype that we put together. And after getting some pretty good feedback, we went ahead to continue development of this Atlas browser into a production quality product. So just to give you a bit of a reminder, how this came about is basically Melanie's group has received the data dictionaries from the cohorts, did the harmonization, and so that harmonized data is loaded into the browser, so users can look for cohorts that meet their specific requirements. So a simple use case is someone may be looking for a cohort that record blood pressure measurements and also have genomic data. So that's what the Atlas browser allows you to do. So now this Atlas is actually live. It's available, publicly accessible. So this is the URL. And more specifically, there are a number of things we did sort of in the background. You may not see much difference from your end user perspective, but we put in a lot of work to make this a production quality product. What that means is now this is a solid foundation for us to develop new features, to build on it as we understand more the needs of the consortium. The deployment of this atlas is automated. That means, if we decide to go to a different cloud provider for hosting, this can be easily done. Back in May, you saw mock data that we made up for 66 cohorts. That was for demo purpose to get a sense of the usability and the functionalities. But now we have removed this mock data, so hence we can actually make this atlas publicly accessible, and we've put in the real data from 5 cohorts. So I'm going to go ahead and show you this browser. Now, I think usability is pretty intuitive during the search. So I think I'm going to spend a little bit more time to show you the data. This is real world data from the 5 cohorts. So as you see on the left here, there are a large number of categories we've placed the metadata into. I'll make a comment on that a little later. But as you expand each category, you can see that, for example, in socio-demographic, we now have some very interesting data. This is all harmonized from just 5 cohorts. Just from 5 cohorts, we are starting to have a very rich data set here. And then we have physiological measurements. So as I said before, blood pressure, maybe it'd be something you're interested in, so we'll come back later. There's now healthcare information, slowly building up with physician practitioner data. There's also non-pharmaceutical interventions along with medication data. So whether there are drug response being collected, if there's administration of the drugs being recorded, that's all now collected. And we also now start to collect disease information as well. So some of these cohorts may be disease specific, or some may just be population cohorts, but disease information about the donors are starting to emerge. And there's also lifestyle and behaviors that we're collecting. And then there's also the more administrative kind of data about the lead PI, enrollment sizes of the cohort, sample data that is being collected. So for the biosample types, we actually have got a graph here as well to give you a sense of what biosample types are collected in some of the studies. So for example, if you're interested in blood, you can very quickly see, oh, these 3 studies collect blood samples. Or on the other side, you're interested, well, what does the Africa Health Research Study collect? And you see that it has these 4 different sample types being collected. So we try to have some visuals to help users, and we can definitely gradually add these kind of charts and interactive information. Coming back here, we also have some more administrative attributes. And then lastly, there are 4 categories that may be of interest. You want to know whether there's clinical data, genomic data, environmental data, or biospecimen data being collected. So this is a yes or no. And lastly, if you want to know what country this cohort is being run, you can get that information. And also lastly, the name of the cohorts. So just to illustrate the search capability, I'm just going to use that previous use case I said. I'm interested in cohorts that collect blood pressure measurements. So going back down to physiological measurements. So blood pressure, I'm going to check that. And I want these cohorts that have genomic data. So going back up, I now go to genomic and check yes. You see the queries are up here, so you can keep track of what you've been searching for so far, and you will find 2 cohorts that meet those criteria. So this is the search interface. It's fairly easy to use. Please do test it out now that it's public, and do give us feedback. One comment I'll make is you probably notice there are now a lot of information on this just one left side. So maybe it's a little hard to scroll or to get to them. And I think this is where we can reorganize the data. At the beginning, we didn't know how much data or what categories of data we're getting. Now that we have a better idea, we can definitely redesign this, reorganize it, so it'll be easier for users to get at the attributes they're interested in. So with that, I'm going to hand the presentation to Melanie. What you've seen so far is the final product, but Melanie is going to talk about how the harmonization took place to get to the browser data, and I'll be happy to take questions at the end.
Thanks, Christina. Yes, so today I'll give a very, very brief summary of the harmonization. And I wanted mostly to talk about, lesson learned and the new tools we have deployed as well, and where, we need your help and the help of other cohorts. So on the next slide, please, Christina. So just a very short reminder, we're trying to bring together the heterogeneous metadata from the individual cohorts and provide that integrated view over that integrated data set so that it can be presented in the atlas that Christina just showed up. So since the last presentation in May, so in addition to the production release to the atlas, so we've added more cohorts. In May, we had only 2.5 mapped. We now have 5. We've deployed a cohort registry, and I will show you in a minute what that means. We also deployed a pipeline that allows the domain expert, the cohort owner, to actually submit their data and how that trickles down into the atlas. And then the whole workflow has been automated. So if you submit to the registry, everything gets propagated and is being made publicly available in the atlas down the line. On the next slide, please, Christina. So the overall use case, as Christina described, a researcher is looking for cohorts that have a specific type of data, for example, blood measurements. And when we're trying to do that, we have 2 types of issue. The first one, which is probably the easiest one, is the syntactic one. So we get, cohort in a bunch of formats. We get PDF or Excel spreadsheets. We get different availability. The licensing, it's not always obvious. The second one, which is probably the hardest problem to solve, is the semantic harmonization and standardization. So different cohorts use different type of attribute and different value for those attributes. And the question is, if I'm looking for blood measurement data, well, I'm probably interested in knowing that COGES has information about glucose in fasting condition measurements, and also that the Goodhand study, for example, collects blood pressure in the left arm, and I think this is diastolic blood pressure. But this is not obvious for a computer how to bring those things together. On the next slide, please, Christina. So this is really the summary of the process we've developed. So the user comes into the atlas with a specific use case and query, and then the rest happens in the background. So we have on the right-hand side at the top, we have a metadata model, which we've formalized as an ontology. We import the cohort dictionary, and we bring those 2 together, doing that harmonization step where we're mapping things, where we are trying to use semantic toolings for text minings and so on to bring those 2 together. Then we load this into a cohort registry, and I will just show you what it looks like a little bit later. We import cohort admin data from the IHCC website. So there's a large table of CSV data with the admin data, where the PI of the cohort, so the country and things like that. And we bring that, we message all that into shape, and we deliver that to the cohort atlas. On the next slide, the lessons we've learned from this pipeline, there are pain points, and I did mention those in May. Those were where we were a little bit stuck, and why in May we had only a couple of cohorts loaded. Specifically, it's hard to handle different type of data coming from different places. So we'd like to have a standard format. CSV sounds like a good choice here. Everybody knows what a spreadsheet is. It makes it easier for the domain experts to work with. And the second step is the mapping. So the first mapping we've done were entirely manually done by my colleague, Becky. So she was really, moving things across one by one and trying to align things. And we've made progress on those aspects, trying to text mine and use existing tooling. And I will tell you a little bit more about that as well. On the next slide, please, Christina. Of course, none of those would have happened without the support and the contribution of those existing 5 cohorts. They've been really instrumental in both building the atlas and reviewing the data before we released it publicly. We were lucky to engage with a set of diverse cohorts, and I believe some of those owners are on the call today. So really, thank you for your help and your support for this project. On the next slide, the cohort registry I mentioned a little bit earlier. So once the cohort data dictionary is provided, we are trying to load it in a standard way, and we've used all the ontology web language to do this. I'm happy telling you more about why, but we thought it was the best in our case, and that this gives us unique IDs for each of the attributes. It gives us versioning. We have everything hosted under GitHub. And more importantly for work, it gives us interoperability with some of our semantic tooling that we already have in-house. So this really fits in well with the suite of tools and capabilities we already have and allows us to develop quickly. On the next slide, Christina, please. So one aspect I mentioned earlier is the difficulty in gathering the data and putting it into a consistent format and doing that semantic harmonization, bringing those attributes together and doing that mapping. So since May, we've developed a pipeline that enables direct cohort submission, where the cohort owner can upload their data directly to that cohort registry I just showed you. And as said, we're using a CSV format. This triggers an automated pipeline, which will suggest mapping. So we're using text mining to suggest mapping. The cohort owner will really, they're the people who knows the best their data and who really can tell us whether it's right or wrong, can review and update that mapping interactively. And once it's all approved and everybody's happy with the status, the file is converted, committed, loaded into the registry, and then freeze triggers the update of the browser down the line. So this is all automated and very reactive process, thanks to Christina's team as well. On the next slide, I just really wanted to give a short idea of what that would like. So this is a test we've done with a little sample cohort. So this is my colleague Giselle Carey from EGA. So you can see that there's a spreadsheet to enter a little bit of cohort metadata. We're asking for a license, which is also very helpful for future use of the data. And then there's a spreadsheet to enter your cohort dictionary. So you can enter the attributes that you're currently capturing. You can add the definition, and we can also use that. So this is the values we're trying to load and map for harmonization and presentation in the browser. Once you've entered your information as a cohort owner, you submit those spreadsheets to the pipeline. And on the next slide, an admin on the IHCC side has access to an admin console, which again is very interactive. The console allows to monitor the process. You can open and links to the spreadsheets. You can trigger the further step. You can run the validation. Everything's versioned. We're using GitHub in the backend, so you can always go back. It's fairly transparent. And once the file gets pushed and committed, everything is reloaded in both the registry and the atlas. You also have a history of the tools that are being run. There's a console at the bottom. So it's really meant to be interactive and transparent as much as possible. On the next slide, Christina, please. So what does the mapping output look like? So you can see on that small EG example, the first column, we generate unique term ID automatically because we use identifier throughout. We propose a mapping in the middle column, and we also indicate why we think this is a good mapping and what our confidence is in doing that. So we capture both the provenance and the confidence score for each of the mapping. The cohort owner can review those and can decide whether they're suitable or not. So for example, in this case, the last one is perhaps less good. We're saying we want fasting glucose level, and the mapping is urine. We can open a drop-down menu. We can see that the next best one would have been blood, and then there's also the rest of the categories are offered. So the cohort owner has the opportunity to review and validate the mappings that have been done automatically. Another interesting aspect is that as the mappings get refined and we get more of those, this feeds back the mapping pipeline itself. So the more data we get, the smarter we kind of get in the next round. And that's done on the next slide, Christina, via existing suite of segmenting tools. So we have tooling for ontology annotation, so that this does entity recognition. We have tooling for ontology cross-references. So if your cohort is already using standards for annotation, such as ICD or SNOMED, we can use that to provide the right mapping. And we are using the ontology lookup service for searching for terms and API availability. On the next slide, please, Christina, the next step for, I think, both Christina and myself is expanding the content of the atlas. And we really want to work with you, the cohort owner, and supporting you in submitting your data to the atlas. There is a little bit of work to be done in terms of the mapping, but we're happy working with you one-on-one to make that as easy and painless as possible. And we also want to explore future opportunity for use case-driven development. If you were on the call on Monday, John Chambers mentioned adding samples link, or Sarah was doing, Biomarker was doing interesting work around mental health and harmonization there. So these seem to be, good use cases and a possibility for further alignment on the data harmonization side. And with this, on the next slide, I would hand over to Thomas, please.
Hi there. Thanks, Melanie and Christina. It was really great to see the Atlas is now live and available, and we would really welcome getting more of the IHCC cohorts to be added to the Atlas, and it's great. We have a process for that now, which we absolutely didn't have in the main meeting. So for any cohorts that are interested in getting their data dictionaries harmonized and available and searchable in the Atlas, then please reach out to us. Christina, if you go to the next slide. So I guess, by starting with the Atlas from the data team, we really just focused on the discovery problem, and that seemed like the obvious place to start for our team. And it is realistic to say, I like David Geiser's analogy in the chat, which was, this really is a list of the books in the library. You can't actually go and actually start to look into the content yet, or the values. The obvious kind of next steps for the data team is to start to think about how to make cohort data accessible, how to start to think about being able to do cross-cohort analysis, and there will be a lot of factors that we need to take into account, like just the size of the data from cohorts, especially as many of cohorts start to generate genetic data, genotyping, genome sequencing. That's the data sizes really start to, for these very large cohorts, really start to get quite significant, as we're seeing through things like programs like All of Us and UK Biobank and others. Things like jurisdictional restrictions, there may be restrictions on the borders that cohort data could cross, and then we have to start thinking about federated models where bring your analysis to the cohort data itself, maybe, and in this kind of potentially in this model of trusted research environments, that we start to see the emergence of those, and we really have to think about what will the IHCC data platform look like. Would it be federated? Would it be centralized? Would it be some sort of hybrid? We are starting to see that the emergence of the technologies that really can scale, particularly cloud-based technologies, and the interoperability standards to enable cross-cohort sort of integration and access, and everything that needs to happen around that to enable cross-cohort integration analysis. And the Atlas is just the first step on down that road. we really want to leverage, we are seeing the emergence of cloud platforms for doing cross-cohort analysis. They're starting to appear from various cross-cohort initiatives, and we really want to figure out how can we work with those platforms, perhaps to leverage those platforms, as we think towards building this IHCC data platform. And I think it's good to see that IHCC is now starting down this process of developing a strategic roadmap. I think we kind of, for the data team, we got something up and running quickly. We've got it's great to see the Cohort Atlas is now a product and available. But I really want us, and then Philip and I want us to really think about, what's going to be the strategy for for building the data platform for IHCC. What will it look like? What will be the model for data access? And we have to really understand what the use cases are, and really then start to reach out to the cohorts and see what their plans are for enabling access to their cohorts. And really the goal here is to really increase the reuse of the cohort data, and that ultimately that will benefit everyone, all of the various stakeholders, be it the cohort owners, be it the research community, and ultimately start to deliver on some of the really kind of grand kind of goals of IHCC, which is to really leverage these large cohorts to make a real impact on biomedical research and healthcare. So I think we will be over the next 6-12 months working with the IHCC leadership, is really to start to think about what will the data platform, what will be the roadmap for this IHCC data platform as we go forward. And hopefully we'll be able to get as much input as possible in that process. So these workshops we're having on Monday and having today, to starting to gather those use cases and input, I think will be really valuable. And I think we'll be certainly reaching out to various people in the data team as we start to engage in this road mapping activity. And that's it. I think have a few minutes for questions. I see there's quite a bit in the chat. Oh, I should acknowledge, here's all the people involved in, this is really the whole set of people that are involved in getting the IHCC Cohort Atlas off the ground. And it's not a trivial exercise, and just solving the Cohort Atlas cohort discovery problem, that's still going to take a number of years. As you see, we've only got 4 or 5 cohorts in the Atlas, and we really want to scale that, but then we also want to think about the wider problem of building some sort of platform, or figuring out what's going to be the model for enabling access to the actual cohorts and enabling that cross-cohort analysis. Great, we've got a few minutes for some questions if there are some. Quite a bit of good discussion actually in the chat. Is there, let's see, Melanie, is there any questions that maybe particularly are worth discussing that maybe we're...
I think Hákon had a good question with respect to the future of the Atlas. Specifically, is there an opportunity to analyze the data within the system, or how do you connect the Atlas to the data?
Yeah, that's kind of that's essentially what I was alluding to as right now the Atlas is a list. It's a list of what's available in the cohorts, a list of what metadata they have collected. That connection to actually accessing the data and all the technology and access criteria and all that kind of infrastructure. That's not something we've looked at, but these are the obvious next steps, for the platform, and to really make the Atlas an entry point to getting that sort of wider access and doing the downstream analysis. So the Atlas right now doesn't have any value, doesn't have the values, and that's a whole other activity that we'll have to think about how best to approach that problem. I mean, we're working on that in other contexts, other projects, in Europe with the Sinica project that I'm leading. We're essentially tackling all of those downstream issues about access and leveraging all the GA4J technologies and working with a set of cohorts to try and get, to work with them to get the implementation of the access technologies on their side. And then same with Philip in Canada. And yeah, that is the obvious next step, I think, as we build towards the data platform.
Yeah, I'll just add to that. So we've been talking with a number of potential collaborators and partners about what that next step is. Thomas and I have always thought about this in the context of a federated model where data could potentially stay home, but there may be other opportunities. But one of the first sorry, it's almost daycare time here. So one of the first things that we're going to have to do is see what we can do in the context of the cohorts and their rules.
Hi, may I ask a question to Thomas? I wonder if you have thought about having a two-stage level of access where people could do some basic analysis on data right in your data platform without downloading any data. And then if people need deeper access, they can move to a next level, and then maybe the owners of the data could get involved in providing access or go through a different procedure, but having an easy way to do some queries in place in your platform maybe stimulates because I from my previous experience, some people in charge of cohorts are not that willing to give you a copy of the data, but are willing to provide you a way to do analysis on their data very easily.
Yeah, it's all very relevant questions, and I think that is what we, there is analogies to this, things like the GA4J sort of beacon analogy, where you can kind of ask basic questions about the presence of genetic variants, maybe without actually getting any information beyond yes or no, and then that gives you, perhaps that gives you the kind of hook in to say, okay, I want to get access to this cohort because I know that there is these set of genetic variants that are really of interest to whatever disease you're studying. There is models for how this might work, and I think, yeah, these are the exact kind of questions we want to try and figure out, and we will need to get, we'll need to get buy-in from the cohorts about what sort of queries they're comfortable with making available in that sort of anonymous, or perhaps maybe it could be like this registered level. We have this concept in the GA4J, sort of this emerging passport technology, where you have an identity that's say verified by your institution that you're affiliated to, and the cohort, the cohort can see, okay, this person, is from this institute, okay, so they'll get this certain level of access, and maybe they can do slightly more advanced queries, and then maybe they want to get full access to the cohort data. They go through a full access application. I mean, it's trying to get this, this level of access right, and we need to bring the cohorts. We can build all the things. We can build all these fantastic things, but we have to bring the cohorts along with us here, I think.
And also, you will need the information about how much informatics infrastructure the cohorts already have in order to support a beacon. You already need to have your own database. So I wonder if IHCC has already collected that information about how much capacity each of the cohorts have in terms of providing access to their own data. Because a different model would be the geo model, where people just upload data with some specific structure and metadata to a common repository, and then that institution is the one that does the querying and the access without requiring the cohort for having their own servers and dealing with all that's needed.
Yeah, well, this is what I was saying, that, what will the data platform look like? I mean, some cohorts may have really advanced their own sort of cloud platform sitting right beside their cohort data, and maybe that's the right way for getting to doing analysis on that cohort. Other cohorts, obviously may not have that advanced IT infrastructure, and we may end up having some sort of IHCC environment that the cohorts could put their data in and have all of the control, obviously retain all the control about access and everything else for the cohort data. But they get the advantage of having an analysis platform sitting beside their cohort data that then could be integrated with other cohorts to do sort of more advanced cross-cohort analysis, which then ultimately gets the reuse of the cohort data, whilst also maintaining the access control with the cohort owners. These are all questions that need to go into this strategic planning activity, I think.
I mean, there are some challenges ahead for some cohorts in some regions. Putting something in a more centralized or cloud-like activity will be amenable or feasible. For some cohorts, there may be some legalities that we'd have to try to understand, or that won't be able to be the solution. And then it may be more of a challenge in terms of having a centralized site for doing data analyses and so on. But it's unlikely we'll be able to identify as one solution fits all type of model, I would think, going forward. But we're going to have to work around that.
I think we're just about to time, so I guess we'll hand over to the next group, or Peter, or whoever's doing the handovers.
I think we're going back to Peter at this point.
Hi everyone, it's Adam Butterworth here. I'm going to be chairing the next session. Thank you to the Atlas team for getting us off to such a great start. Fantastic to see the progress and to hear the discussion there. And I think clearly some really valuable input that we should continue to seek. Hence the value of having this kind of meeting, even in a virtual format. So I'm here in Cambridge in the UK, where I have the pleasure to co-chair the Scientific Strategy and Cohort Enhancement Working Group with Hákon in the US and Geoffrey in Israel. So if we could go to the next slide, please. In this session, which is about the next, 75 minutes or so now until the break, we're going to hear about the progress that's been made on the scientific agenda. We heard, for those people who joined on Monday, about the COVID pilot projects that have kicked off in the areas of mental health, biospecimens, and South African Host Genome Initiative. We're going to hear from Eric about the process of awarding the funding for the initial pilot projects, and we're going to hear from 3 representatives from the other funded pilot projects that are just beginning, to give you a flavor of the scientific projects that have kicked off. We're then going to hear from Hákon as well, firstly about an update on the previously funded pilot project on polygenic risk scores, which those of you who attended the last meeting will recall. But also about a new exciting opportunity to engage potentially large swathe of the IHCC and the Davos Alzheimer's collaborative area as well. Next slide, please. So the theme for this session was really around updates on Objective 4, which, as Peter already alluded to, is really about engaging as a broad swathe of the IHCC membership, trying to get as many cohorts as possible involved in developing the key topics for our scientific agenda. And the various key actions and next steps that came out of the last meeting, ICS 3. So we talked a lot on Monday about the need for the roadmap, which we've just heard about in terms of the Atlas and the data side. Obviously, we need an important component of that will be the scientific strategy as well, and that's part of this meeting and the plan going forward for the new scientific committees. We had an action about joining the Davos Alzheimer's Collaborative and some of the initial calls, and we'll get an update in more detail on that from Hákon later, but thank you to all those cohorts that did participate and express an interest. There are now about 30 IHCC cohorts that have expressed an interest in engaging with that initiative, so that's got some excellent momentum. We planned that the scientific strategy team would try to launch somewhere from 3 to 5 scientific initiatives with a 12 to 18 month time frame, to demonstration pilot projects, if you like, to try and show the value of IHCC and to try and galvanize cohorts into some actual science. And we're pleased to tell you about the 6 projects that have been approved now for funding. Because of the COVID situation, we decided that we should try and collaborate on COVID-specific activities, and you've heard about some of these previously on Monday. And that, of course, through implementation of the charter and the new governance structure, etc., and virtual working group meetings, we should continue to develop a shared scientific agenda. Of course, that's part of the agenda for today's meeting and the breakout sessions later on. So that's the actions and where we are with those at a very high level. And now I'd like to turn to the more interesting bit, which is hearing some details of the science that's unfolding. First of all, if we could start with Eric, please, on an update on the pilot project and the funding process. Thank you.
Thank you, Adam. Can you hear me okay, everyone?
Yep, you're good.
Good, thanks. Okay. Yes. Next slide, please. So move into where we have come from since the last meeting. So just a quick look at the timeline and the process that we went through to meet this objective of funding a number of scientific research projects. So essentially, we went from zero at if you look at coming out of our May summit. To funding projects in approximately 6 months, half a year, and really going from zero when we had just the concept coming out of the summit, and going through the timeline over the last 5.5 or so months. We started with a proposal solicitation, which was about mid-June. That was put together by the scientific strategy team as in terms of the requirements for submission. And the FOA was issued across all cohorts. In July, we, from feedback that was received from questions and kind of refinements to the process, we put out an FAQ just to address certain questions that were some commonality there, as well as some budget guidance documents and some updates based on those questions. A couple of weeks later, we did put out a call for volunteers to participate in the scientific project subcommittee, which was very successful. I'll go through some more details of that in just a moment. Mid-August was the proposal deadlines where we received the submissions. Within 2 weeks, we had convened the scientific project subcommittee to review those proposals. And within a week from that point, we did have conditional selections were complete, and were notified those that were awarded or were conditionally awarded the funding. Since that time, and again, going from zero to funded included having to develop all the required infrastructure, terms and conditions, reviewed by our legal counsel to make sure that we were representing not only the interests of the IHCC, but also that we were honoring our obligations to our funders and making sure that that transfer of funding met their requirements as well. So there was quite a bit of back and forth to make sure that we were contractually meeting our obligations and transferring those to the awarded projects. And happy to announce that just this week we have begun issuing the contracts for review to those awarded projects, and so there's probably several of you on the call who have received or will be receiving shortly those contracts. So that process has begun, and which will lead to our kicking off of the projects, by the end of the year. So just quickly, thank you very much to, we had a very positive and overwhelming response. We actually had additional volunteers. We did narrow it down to this select group based on a couple of factors, including, we had very good representation, not only from cohorts, but also from industry, our industry partners and funders as well. We did remove those that actually had submitted proposals as well, just to keep a completely unbiased representation of the review. And the review process was done via survey. Great contribution. And the feedback and the analysis of these projects. On the merits of the submission was really, really well represented. It was co-chaired by Katerina Ferrecchio and Jeff Ginsburg, and they led this process, and I think we had a very, very positive result. So the results being, we had a very, very wide variety of global research topics. You saw yesterday the 3 that were channeled out of the COVID-19 emphasis that we had in the summit, and we'll see here just when I'm done here, we'll see the next 3. So we did have out of the 7 proposals that were submitted, we were fortunate enough to have funding to provide 6, and we did put a cap. I didn't mention it here, but we did put a cap of for these pilot projects at 150,000 US dollars. And we were able to fund 6 projects, 5 fully and 1 partial. So there was some remaining work, and we were able to, you heard from Michelle Ramsey yesterday, we were able to support that, and they had supplemental funding from Illumina that enabled them to move forward. So that was a very fortunate and good result there as well. So just in terms of participation, the average number of cohorts per project that are participating is 8, with a range of between 3 and 18. Again, very positive there. And in total, we were able to, thanks to additional funding, which, by the way, coming out of the Atlas discussion, we had made a presentation. The Atlas team had made a presentation to Chan Zuckerberg, and that resulted in an additional about $170,000 of funding to pick up an additional project in full. So we were able to actually pick up that additional project. So in total, we're looking at $800,000 in US in total funding. If you look back and include the PRS project that Hákon will be updating shortly, we're approaching $1 million in funding, which I think is a milestone we should all be very proud of. And again, thanks very much to our funders for making this happen. And this is where we get to prove our merit, and it will set us. The success here will set us on the right path, and helping develop not only our strategy but our ability to secure funding to move forward. And next slide. That's it for me. Any questions about the process, or any questions about the process overall, or the results? Comments?
Eric, can I just, thanks for the update and the presentation. Can I just make a comment, which is that, thank you to the people who are involved in actually putting the process together. And because of the short time frame for the funding, I think in some way we were a bit back to front. We had to kind of think on our feet. And I know there was a lot of hard work that Eric and his team had to do to, as he mentioned, to ensure that all the funder requirements, et cetera, were satisfied, and to put in place at short notice the financial details of the funding call. I believe we saw on Monday that there's a plan to put in place an SOP for this, for kind of future pilot project funding rounds. Is that right, Eric?
That is correct. And I'd be remiss if I didn't also thank, we brought on board a new resource on the Secretariat, Waitina Mangum, who I don't think is on the call with us today, but she's taken on the burden of managing these contracts, which again, her 0 to fully funded started really in September. So it was really sort of a Herculean effort for her to come on board so quickly and make this happen. So a special thanks to her and Teji for bringing her up to speed so quick.
I see a question from Jeff in the chat is about knowing how many cohorts are involved across all the different projects. I suspect that's a number that nobody has off the top of their head, but perhaps we can dig out during the remainder of the day to get an answer later on. But that would be important for us to know clearly.
Yeah, I can pull that together. Yes.
Yeah, one other thing I just wanted to comment. First of all, thanking Eric for his leadership in this process, and also the review committee, I think, was as good as many study sections I've been on. I thought the engagement and the level of critique and review was outstanding. So I think that bodes very well for future endeavors of this type.
Agreed. Yeah. Thanks to everyone and to the people who put proposals together as well. And we're only as good as the scientific ideas we come up with. So important that we try and engage as many different cohorts and investigators as we can as we go forward into hopefully future rounds too. So we're bang on time. So we'd now like to go to the presentations that give an overview of some of the pilot projects that kick off. And I think first on the agenda we have David Hughes.
Thank you very much, and thank you for the funding. The IHCC, it's been amazing actually work from you to get the funding call out and reviewed and back to us so quickly. That was very impressive and great to be funded. So if you just put up my slides there. Yeah, got it. Thank you very much. So So the project is exploring the role of genetically determined BMI in infancy, childhood, and early adulthood on colorectal cancer development in later life. And I'm here located in the Conway Institute of University College Dublin in Ireland. And if we move to the next slide, please, Adam, you will see this just as a slide, is it just an interest in this book that I've just started to read, which really shows the power of longitudinal cohorts. Just wanted to mention it. This is written by 4 preeminent psychologists, and they give a nice overview of what they've learned about how humans develop after tracking the lives of thousands of people from birth during their life. And it's usually, the book is really put together mainly data from such cohorts as collated together in the IHCC. And of course, it shows the influence of genetics, which is important for the project that I'm going to present, but we're not subject to such ironclad laws, but instead it's a matter of possibilities and probabilities. So very, very difficult for us all to tease out, but really, really interesting. And discoveries such as put together in this book will promise to help schools, parents, and all people foster well-being and ameliorate or prevent developmental problems. So just as a background to this, it's also a nice review on this in Nature, which was a very apt cover as well, because it showed an article on Viking history, which is very, very pertinent to me here in Ireland, as those guys certainly seeded a lot of the genetics carried around by modern Irish people. So on to the next slide, which is back to the project then, and just wanted to thank my applicants, of course, without who this work would be impossible. My co-applicant, Mahdi Sheikh, in the International Agency for Research on Cancer in Lyon, in France, where I worked with him for a number of years as well. And we worked together in many projects. And then also Veronica Fiduco, who is currently at Emory University in Atlanta, but will be moving to MD Anderson in Texas in the beginning of the new year. And also then we have some collaborative input in this project from Neil Murphy and Heinz Freisling in IARC as well in Lyon. And for this project, we will be recruiting a postdoc for a year based at IARC and also at UCD to work on this project specifically. So this is just a little bit of background on specifically on colorectal cancer and the risk factors. So in general, worldwide heterogeneity in cancer incidence suggests the etiological involvement of modifiable risk factors such as obesity, overweight duration, and weight gain in adulthood with cancer risk, particularly obviously obesity-related cancers such as colorectal cancer. And obesity, most likely through the nexus of genetics, diet, and lifestyle, is strongly associated with colorectal cancer development. This slide shows on the left-hand side a little summary from the World Cancer Research Foundation for the best evidence risk or protective factors for colorectal cancer. And then in the middle of the slide, I've added the likely biological mechanisms through which these risk factors are mediated, such as inflammation and metabolic syndrome. And early life exposures appear to impact obesity in childhood, and a concomitant influence on cancer incidence is plausible. For example, dramatic increases in colorectal cancer incidence observed at younger ages in many world regions that may relate to early life exposures and higher obesity rates in younger generations. But this link is not established, and few studies have attempted to assess life course obesity exposures and cancer etiology by consideration of genetic predisposition. Both adult and childhood obesity have strong and partially overlapping genetic determinants, whereas those for infant obesity may be somewhat distinct. In all, diverse genetic factors for obesity through life stages does infer different metabolic processes in early versus later life that may underlie adiposity and cancer development in adulthood. Therefore, we hypothesize that varying obesogenic inherited predisposition in early and later life stages infers different metabolic processes that may underlie this adiposity and differentially impact risks of colorectal cancer and possibly for earlier onset disease. So then moving to our aims and objectives in the next slide. Our main aim is to assess whether life course specific genetic markers of obesity influence colorectal cancer development. We will use available data on single nucleotide polymorphisms, so SNPs, so robustly associated to obesity, to construct 4 specific genetic risk scores, so GRSs, for obesity in infancy, childhood, adulthood, and then overlapping childhood, adulthood. These GRSs will be applied to evaluate risks for developing colorectal cancer using existing genetic, anthropometric, and cancer incidence data from available relevant IHCC cohorts. So our agreement thus far to participate is from the UK Biobank, the European Prospective Investigation of Cancer and Nutrition, which Mazda and I work a lot with and are working group members, and the large International Genetics and Epidemiology of Colorectal Cancer Consortium, or GECO. And GECO includes quite a few of the also a few of the IHCC cohorts, as well as other cohorts that are not currently in the IHCC. So additional IHCC cohorts with pertinent data are also invited to join. Especially, we would really like to increase the ethnic diversity, although we do have quite a diverse network within GECO. But for example, we would like to have more involvement, say, of Latin American or African American or African cohorts to be very welcome. We do realize there's a problem with cohorts having available GWAS data that we can use. And I really like to see the presentation earlier from Christina on the IHCC cohort atlas, which would be very helpful for looking through these things. So then moving on, just to summarize the cohorts that we will use, the slide outlines the main research questions that I have just sort of discussed on the left-hand side, and then the cohorts that we will examine these in. So just a background on those cohorts, the UK Biobank is about 500,000 people that was recruited over 15 years ago across the UK. And they have so far got nearly 6,000 colorectal cancer. So there's extensive information, including BMI, body size parameters in early life and adulthood, which is critical for our project, and existing GWAS and clinical biomarker data for all participants. And GECO is an international collaboration of prospective and case-controlled studies focusing on genetic risk factors and gene-environment interaction in colorectal cancer. We've got GWAS and epidemiological data for over 130,000 participants from 70 studies, North America, Australia, Asia, and Europe. And that's led to over 50,000 colorectal cancer cases having accrued, so giving us high statistical power for our assessments. And then finally, EPIC is a large Western European prospective cohort of 10 Western European countries with roughly over 500,000 participants, 2/3 which are female, that were recruited between 20 to 30 years ago. So far we've got 2,700 incident colorectal cancer cases matched to equal number of controls with GWAS data and biomarker data on about 1,500 of these for inflammatory and metabolic dysfunction, nutrients, etc., which are important for our mechanistic explorations. And then on to the next slide, please, which is really summarize our analytical strategy. So we will use available data on SNPs robustly associated to obesity. So these will be identified from published GWAS and gene phenotype databases. Mainly we're looking at ones defined for BMI. We will extract these SNPs or their proxies from the UK Biobank, EPIC, and GECO to construct 4 specific weighted genetic risk scores for obesity in infancy, childhood, adulthood, and overlapping childhood, adulthood. The GRSs will be calculated from the sum of risk alleles corresponding to obesity phenotypes in each individual and weighted by the effect size estimate of each genetic variant according to the strength of its association with the given trait, for example, obesity in childhood. Then we will apply that to evaluate risks for developing colorectal cancer and also assess whether the associations differ by age, sex, and select modifiable exposures, such as lifestyle or dietary factors, anthropometry measures across life stages. So cause-specific risks for the GRS cancer associations will be estimated using logistic regression models or Cox proportional hazard as appropriate. We will further test these to explore the role of major underlying mechanisms for colorectal cancer development, for example, metabolic dysfunction and inflammation, using existing biomarker measures in the UK Biobank and EPIC. And also where they're available in other applicable IHCC cohorts, that would be very welcome. The figure in this slide shows our causal framework relating early life obesity, assessed as BMI, to later life colorectal cancer development through direct red arrows and indirect effects, which are showing in the light blue arrows for our different genomic risk scores across the life course.
David, we're hitting the 10 minutes.
Okay.
Do you have any more slides?
Yeah, nearly finished.
Okay, great.
Go on to the next one. It's just our intended impact. So we're nearly there. So just to say that if you, this project will add pertinent knowledge linking genetically defined obesity and colorectal cancer development at different life stage. And hopefully this will inform cancer control strategies and public health policy, strengthening the evidence base for windows of opportunity interventions to reduce obesity and prevent colorectal cancer, and also to provide pilot data for refining the genomic risk scores and a larger study on obesity-related cancers and survival after cancer diagnosis. And thank you very much. Any questions are welcome.
Thanks very much for kicking us off, David. Great to see the project outline and to see that there's possibility for other cohorts to engage as well if they have the relevant data. I suggest we use the chat function for to provide questions for David, and he can answer them as they come in. I think in the interest of time, we'll move on to try and stay on schedule, please. So if I could ask Arash to speak next, thank you.
Hi, everybody, and thank you to IHCC and the funders for this opportunity. As you could see in the printed schedule, in the PDF schedule, I was asked last minute to replace Andre, who was supposed to present on behalf of the PIs of this study. That's why I didn't have time to shave. So that's the headshot is different. So the next one, please. The project I'm representing on behalf of all the PIs of the study is a pilot study of high-throughput metabolomic biomarker measures in diverse ancestries with 4 co-PIs from different institutes and team members also. So as a brief background, as you know, chronic diseases impose a high burden on health system in different parts of the world, and health outcomes are really significantly improved if you can do early diagnosis and intervene at early stages of the disease. The problem in most cases is that early diagnosis is very difficult, and particularly individuals in low and middle income countries or the minority populations and even in high income countries do not have access to such early diagnosis and intervention facilities. So one of the ways to move towards early diagnosis is metabolic profiling, which is a highly scalable model for risk prediction, and particularly since relatively it's lower in cost compared to many other methods used in precision medicine. So it's a route basically to individualized medicine for these populations with lower access to those facilities. So this study was developed with the first aim being to develop a structure for the metabolomics within and developing a strategy for coordinated metabolic screening for all of the IHC. So it's a pilot study to showcase the development of a structure of different cohorts, different populations coming together to have this kind of metabolomic profiling on the populations they represent. And in addition to that, the second class of aims is to generate these data on about 5,000 individuals, given the funding that we have. And the genetic, and correlate them with genetic or health outcome data, and also now analyze the association between these metabolite levels, such as lipid profiles and genetic data. So there are 2 sets of aims, as you can see. One is to showcase this coming together of different cohorts, and the second is to generate data that can be used for metabolomic profiling and disease-associated analysis. So we're using Nightingale Health blood biomarkers, which has been used in several studies. Nightingale's panel is an NMR-based platform, and it analyzes about 228 biomarkers. You can see the classes of course, the list of the biomarkers is very long. You can find that on Nightingale website, but it's a very lipid focused, let's call it, panel with lots of lipoproteins, fatty acids, inflammation biomarkers, amino acids, different fatty acid ratios, etc. So it gives a very good picture of the metabolomic profile, particularly when it comes to lipid biomarkers, and 14 subclasses of apolipoproteins are included. And there have been many studies showing how risk score based on this panel can predict different outcomes, particularly cardiovascular disease, etc. So, and it requires about 100 microliters of plasma serum, which is really a small amount, which is feasible for many cohort studies to contribute. So the participating cohorts so far in this study, as you can see, we have a diverse population composition. South Asian cohorts is a study called BELIEVE, which Adam is the PI. This study is giving 1,500 samples of from Dhaka, Bangladesh, South Asian ancestry. ELSA Brazil, which is a longitudinal health study conducted in Sao Paulo, in different parts of Brazil, I should say. There's 1,000 samples from civil servants across Brazil, and Andre is the PI of that study. Golistan cohort study, which I represent, is 1,000 samples from Northeast Iranian general populations, a high-risk area for upper GI cancers. And CHOP, Children's Hospital of Philadelphia, which is giving 1,500 samples, mainly of African-American children. And Hákon is the PI of that study participating in this effort. But as I mentioned before, the project is developed with the intention of scaling up beyond these 4 initial pilot studies. This is an initial pilot to show how we can develop this strategy to combine different cohorts, different populations in this platform. And it also, since other big studies have used this platform in the past, that there's a potential to combine with other IHCC cohorts which have used this platform in the past, such as UK Biobank, Mexico City study, UK Blood Donors Cohort, Kaduri Biobank. Other places which have also used the Nightingale platform can be combined with the data here, in addition to the other studies that in the future can also do the same thing and join this data. We also have some cohort-specific outcome studies. So the South Asian cohorts focusing on diabetes. In Golistan, we are focusing on ischemic heart disease as disease-based study in cohort-specific outcome, and autoinflammatory diseases in CHOP, which is a very good source for studying such diseases. Those are cohort-specific outcomes that we have in mind. Next, please. So this is a tentative timeline, given all that's going on with COVID and the challenges that we are facing. All studies are preparing samples to be shipped. We hope to do it by the end of this year. Nightingale will provide the ready for analysis data by March, hopefully. We'll have some project-specific analysis planned for June 2021. We will, as said, we will report back to IHCC with recommendations and guidelines about how such a strategy can move forward with cohort expansion and other studies that would like to repeat the same experience or have a shared experience. And upon completion of these analyses, we'll make the data available to all IHCC cohort members according to the agreements made. Next, please. And thank you, everybody. This is a reminder of last year's meeting in Iceland. This beautiful scenery that we enjoyed, and hopefully this is a reminder that we'll come together in person real soon, hopefully. And this church, as I understand, has a very difficult name. Hákon can tell me. I forgot, but it was really beautiful. So thank you very much.
Thank you very much, Arash. Hákon, do you want to remind us of the church name?
So Hallgrímskirkja. Hallgrímur is the name of a priest in Iceland who sort of was a major driver of building this. So Hallgríms Church or Kirkja.
Excellent, thank you. Well, as you say, Arash, hopefully there'll be future beautiful pictures from future in face-to-face meetings rather than pictures of Zoom screens. Thanks for the great talk. Does anyone have any quick questions for Arash before we move on?
I have a question about whether you were able to verify the sort of standard operating procedures for the sample storage to ensure that you'll get equally quality data from the different cohorts.
Maybe Hákon, would you like to talk, because you were?
Yeah, I mean, we have not sort of tested that, Geoff, but I mean, we have done, for example, multiple measures of our cohort samples for other biomarkers. So we know our -80 storage for plasma has been good. Same thing with Adam, and I know all of the 4 sites they store the samples at -80. So I mean, I think we just have to sort of hope for the best that was appropriate.
Yeah, and Nightingale has a lot of experience actually in telling cohorts about the properties of their studies. For example, we have a blood donor cohort, and they reported to us that many of the participants appeared to have substantially high ethanol levels in their blood, which was a bit concerning for blood donors. It actually turned out this was an artifact of the enhanced swabbing of the arm with an alcohol swab before the venipuncture, which was being detected in our study but hadn't been seen in other studies. So we didn't have lots of highly intoxicated blood donors, but we did have something that was causing a problem and masking 1 or 2 of the metabolites in the measurements. So we'll work very closely with Nightingale to try and understand the differences between the studies. Yeah, good question, Jeff. Okay, let's move on in the interest of time. And the third scientific presentation is from Mahdi, who's from IARC in Lyon.
Thank you very much. This is Mahdi Sheikh from IARC, and I would like to thank the IHCC leadership and also scientific committee for providing this opportunity for us. And next, please. So this is about the project Opioid Cohort Consortium, or OPICO, to investigate the effects of regular opioid use on mortality and on cancer development. Next, please. So it is about really the global crisis of opioid use. Unfortunately, this crisis is affecting many countries in the world, while the short-term health consequences are well documented. Unfortunately, the long-term health consequences of opioid abuse or opioid use has remained unknown. Next, please. So when we talk about opioids, in 2018, it was used by almost 60 million people. Next, please. It has 2 main categories. The first category, opiates, or the natural subcategory of opioids, mainly opium and heroin, used by 30 million people in 2018. There have been many studies, particularly on opium use, to see whether opium use is associated with long-term health outcomes, mainly cancer and mortality. So here you're showing the results of cancer studies. In human studies, it's been shown that opium use has some associations with at least 9 cancer types. And the experimental studies also well documented the genotoxicity of opium use and carcinogenic compounds in some opium samples. So next, please. As a result, opium consumption was recently evaluated by the IARC monographs program, and it was classified as group 1 carcinogen to humans. Next, please. The other group of opioids are pharmaceutical opioids, and this is the crisis in the world. Again, we went to, I'm summarizing the evidence here. Human studies, we only have like a couple of registry data linkage studies showing consistent associations between using opioids, pharmaceutical, and also several cancer types, including lung, urogenital, and liver. Well, there have been many experimental studies showing some genotoxic toxicity associated with using different types of opioids. Also, the tumor promoting effects of opioids like immunosuppression and neovascularization has been shown in some studies. Also, cancer tissues, some cancer tissues, opioid receptors have been found, and the studies have documented their involvement in tumor initiation and progression. But as you can see this evidence, our classification is only correct about opium. We have now many concerns about what are the carcinogenic risks of pharmaceutical opioids. And we couldn't find any cohort study looking at this outcome because, well, the registry data linkage studies, there are lots of confounding effects and biases from these studies, like confounding from smoking, alcohol, or the indication bias. And we couldn't find like opioid use data. Most cohorts have not gathered opioid use data, and those cohorts with opioid use data unfortunately have, well, the number is very limited that we cannot look at these outcomes like cancer incidence and cause-specific mortality, and also the patterns of use, the legislation, and also the underlying chronic health conditions are different among countries. Therefore, we thought that the best approach is to take a comprehensive consortium-based approach to answer this question. And also, this is just some example of the Goldstein cohort study that has large number of opium users, and it has been shown that opium use is associated with all-cause and cause-specific mortality, and also some results from the UK Biobank show similar things or mortality for opioid use. So just an overview of the Opioid Cohort Consortium. Just recently, this was approved for funding thanks to the IHCC. The overarching aim of this consortium is to build a strong international resource for multidisciplinary scientific studies on the use of opioids and their long-term hazardous effects. So we are building an infrastructure. The main exposure here is the use of two categories of opioids. So some cohorts have gathered information on opiate use, opium and heroin through questionnaires, and some cohorts that have gathered medication use data on opioids, and some other cohorts have linked their cohort data to the registry, national prescription or medication dispensing records. So these are the exposure we're interested in, regular opioid use, and the main outcomes, we have two outcomes of interest, cancer and mortality. So all types of cancer and digestive cancer, respiratory, urinary tract, brain cancers that we found associations in our previous work with opiates, and also for mortality analysis, all cause and cause-specific analysis. So the aims and approach we are taking are to organize data on opioid use from large-scale prospective cohorts. As I said, different cohorts have used different approaches. Some have gathered medication use data, but not specifically opioids. So we need to extract this data on opioid use, which is like more than 20 category of drugs, of medications, and organize them using a uniform protocol. And compile new data on opioid use in large cohorts through linkage to national records. I will talk about this in a minute. And also assess the type, distribution, and extent of opioid use across diverse population and determine the association of opioid use with cancer incidence and mortality. So here is just an example of the cohorts that we have like negotiated with the PIs or some investigators in this cohort. More than 1,700,000 people will be involved up to now in the OPICO. We will go ahead with submitting formal requests to each cohort. Next, please. And you see the diverse populations. So, well, excluding some two cohorts that we still do not have data on opioid, the number of opioid users, but you see almost 70,000 opioid users are among these participants. Next, please. So now we have one thing in this consortium want to do is that we have good national drug prescription or dispensing records in some countries, and having well-designed cohort in these countries give us the ability to link their data and assess the opioid use among these cohorts at enrollment or at some time points. So we're assessing the feasibility of this approach in 2 studies, 45 and up, and also Generation Scotland study in Australia and UK. And also we are negotiating the feasibility of performing this approach in more cohorts, hopefully to create data on opioid use to have a very good resource. Next, please. So the future steps and aims, as I said, to build a resource for studying opioid, which is a growing concern in the world. And also we are, our colleagues at IARC are creating some IARC analytical hub, working on that to give the ability for data sharing and ability to analyze the data anonymously without being able to download the data. Something was very interesting in today's presentations about the atlas. So this is in line with that, and hopefully we can collaborate on this. So some of these cohorts have genetic data available. Some of these cohorts have biosamples available. So the future, we can use this harmonized data for future studies on genetic studies and biomarker studies. Next, please. So, and I'm inviting actually more cohorts. Please contribute to this project. We are looking for cohorts that have data on opioid use, questionnaires or data linkage, some outcomes, vital status, cause of death, cancer, and also demographics, smoking, alcohol intake, and chronic health conditions. So here are the contact information. Please, if you have any questions or if you can contribute to this study, just contact us. And finally, I would like to thank all colleagues from different countries, different institutes that helped us, that are helping us in this project. Particularly, I would like to thank Dr. Paul Brennan and Dr. Hilary Robbins, who are co-leading this consortium. And if there are any questions, I'll be happy to take.
Thanks very much, Mahdi, and thanks for answering my question about what data do cohorts need and how should they get in touch. Fantastic to see presentations where we have solidified cohorts already, 1.7 million people already involved, but opportunities for other parts of the world to get involved as well, which is excellent and exactly what we want to do with our scientific agenda.
Great, thank you very much. Yeah, if cohorts have got medication use data, we can extract the opioid use data. Thank you.
Brilliant. Great to hear. Okay, I'm going to turn over now to Hákon, who's going to finish off this session for us.
Thank you, Adam. You can hear me okay? So
yep, you're good.
If you go to the next slide, so I was going to update on the progress, sort of mostly since our last meeting in May, on the polygenic risk score pilot that we undertook sort of on a short notice at that time. And the DAC, which is the Davos Alzheimer Collaborative that we sort of, have partnered with from the IHCC standpoint and formulated a work group and sort of moving forward with research in Alzheimer's disease, and then close on opportunities for cohort involvement. So if you go to the next slide, and of course, I mean, all of these projects are sort of really directly an extension of our vision of sort of, networking globally diversity cohorts from essentially all possible countries, and sort of extensive sort of collaborative network, if you will, for data sharing. So they are all dependent on this, and that sort of is the driver behind these projects, which is really in keeping with our vision and strategy. So as you recall, we presented preliminary data on polygenic risk at the meeting in annual meeting we had in May. Patrick Slayman was sort of the lead analyst who presented on this. And it was sort of really a proof of principle to demonstrate that we would and we were able to work together across different cohorts and share data and generate results. All IHCC cohorts were invited to participate, but we ended up sort of with about 12 cohorts engaging. This was a very short period of time, only really a couple of months, at the time when this project was launched. And we sort of, did this on the on the terms of federated model, where all sites were guided to generate the sort of summary statistic from polygenic risk that was sort of informed from analysis at CHOP, and the summary statistic was subsequently meta-analyzed together. If you go to the next slide, the key phenotypes we sort of addressed at the time was BMI and diabetes, and we also included hypertension and asthma because these are both common traits. Hypertension, of course, or blood pressure, should I say, obviously being measured essentially at all healthcare visits, so these are data that are available, asthma being very common, and type 2 diabetes similarly, and BMI essentially available for all subjects. And the key here was to leverage existing resources and sort of expertise, and to establish a trans-ethnic polygenic risk across sort of these international cohorts based on summary statistics. Again, the two common complex phenotypes and two traits, and we sort of emphasize one of each here, BMI and type 2 diabetes, which sort of where we have most data, most complete data, and sort of basically based on GWAS data that was present at the sites that participated, and based on public information on polygenic risk that had mostly been sort of published across European ancestry cohorts, at least for BMI, but there were actually some diverse polygenic risk scores based on individual cohort scores that we could leverage to sort of come up with an LD PRED weighted structure. LD PRED is basically an R sort of statistical program that we use to sort of generate polygenic risk based on Bayesian algorithm, and it leverages the LD pattern of the data. And we did this, and Patrick sort of drove this across the African data set, across the mixed Hispanic data set, across the Asian data set, European, as well as all combined, which was trans-ethnic. These were the cohorts that initially participated. So we had, even though that sort of was a dominant US and Europe participation, there were still diversity patients in these cohorts. Now we sort of have been working robustly to try to get data use agreement in place with 23andMe because they have such a big data set. But that has not happened yet, and we sort of decided that we would just move forward and get the paper ready without the data. If we get that data, ultimately we can always drive another bigger paper with more cohorts. But you can see that cohorts from Shanghai, Brazil, Norway, CHOP-based cohorts, which is sort of quite international in terms of how we've collated it, and the nurses cohort study from the NCI, and women's health study from NCI, which is also a diversity cohort. They were sort of the drivers of the initial data. So if you go to the next slide, I think we have sort of the algorithm. This is basically just to show that we leverage the sort of the publicly available polygenic risk scores to sort of and we applied that to a reference data set from CHOP, where we had 2,500 people imputed of African or Hispanic or European ancestry, and we were able to sort of come up with a trans-ethnic profile with this, and then we sort of weighted this into sort of differential fraction of the causative variants across the individual ethnic groups and trans-ethnic. So this turned into sort of 5 × 8 files or 40 files that were disseminated to each site to generate polygenic risk. And they did so and then gave these results back to Chop, and Patrick meta-analyzed these data together. So it was not trivial to get this done sort of in a short period of time, but it sort of, we managed to do that. So if you go to the next slide, I think this slide sort of shows just sort of representative results, just to demonstrate that the trans-ethnic score was actually even more powerful for the various different sort of fractions or causative variants than the individual sort of cohort scores. So, for example, representative here from the Shanghai men and women's study, you can see that trans-ethnic score actually outperforms their ASEAN scores across all of the essentially all of the fraction sizes. So this is obviously interesting because, I mean, there hasn't really been any publication yet on trans-ethnic polygenic risk scores, and so that would be really the focus on that first paper, driving it mostly based on BMI and diabetes because the other traits are not quite as well performing or complete. So if you go to the next slide, I think this sort of summarizes that the trans-ethnic score is the unique feature here, which we are just about to finalize now into a paper. And if you, for example, look at the top 1% or the 99th percentile of the area under the curve, for Shanghai, the ability to predict the BMI is 76%, which is actually quite good, and it's no less than in a European, for example, in the MOBA cohort, which is 0.75. And so this approach seems to work, and so if we can get more cohorts to participate, we can then generate another bigger paper down the road. But we will at least drive a paper sort of with what we have, with this sort of trans-ethnic focus, and then of course if we get individual level data, because this analysis was based on summary statistic, we will have more power to sort of generate weighted risk scores across each site. And next one sort of just is to show, I mean, we will circulate this before the end of the month, and the goal would be to give people close to a month to edit and comment and to get the paper submitted before the end of the year. And if we go to the next slide, I think this gets us now to the Davos project, the Davos Alzheimer Collaborative, as I mentioned, or DAC. This was actually initiated in Davos in Switzerland by the World Economy Forum earlier this year. So this is a very recent effort. So it's almost amazing, sort of, Lexit has sort of, managed to walk on with over the last number of months. And this initiative is sort of, from a financial organizational standpoint, led by the World Economic Forum and the Global CEO Initiative of Alzheimer's Disease, which George Radenberg sort of, spearheads. And there are multiple other sort of, thought leaders sort of in that organizations. And they have, since DAC was founded, they've established multiple collaborative partnerships sort of with other organizations. And as you recall, they presented at our meeting back in May. And we sort of, took up an initiative with them and have partnered with them with our cohort effort to provide sort of a global universal networking cohort across different continents to try to drive Alzheimer's research. And if you go to the next slide, so there are three major priorities. The cohort, sort of this mega cohort we reference here as target identification network cohort. I mean, this is sort of really the primary driver that sort of is the foundation of this effort, and it includes multiple sort of large-scale global cohorts from the IHCC. We have actually, as Adam mentioned, 30 cohorts that have signed up already. Half of them have completed a survey so far, delineating what they have in order to support this effort. So that's quite impressive. And then the other two components to this are sort of, a global platform to support clinical trials in the future that we hope will sort of come out of this discovery work that we will drive in collaboration with DAC, and sort of, the healthcare impact that this will have on patients and future patients. And this initiative is sort of really a 5-year plan with the current focus, and we'll start with a pilot effort. And I think the next couple of slides, they describe that. If you go to the next one. The work group chairs for DAC, so I co-chaired this with Elias Cerrone, and as Cerrone, as you know, was an NIH director here for a couple of terms, probably 16 years ago, something like that. But he was also a head of research for and development for Sanofi for several years, sort of prior to sort of semi-retirement. But he is sort of the lead member of this team from the Davos site. And the strategic objective is, as I said, the sort of, focus on improved healthcare outcomes for people living with or who are at risk of Alzheimer's disease, and sort of, with the notion of generating new therapeutics and biomarkers to monitor disease onset and progression and the like. And that has sort of, worked towards establishing key partners. I mean, IHCC is really the lead partner because, the Cohort Foundation is sort of coming from this effort and led by Geoff and Terry. Philip, Thomas, and several other people are involved with that. The Gates Venture is also similarly heavily involved to help Thomas and Phil with sort of a atlas that would sort of be a data hub for this initiative, which obviously would be sort of parallel with what the Atlas is doing, as we heard from Team A earlier, for IHCC in general. So if you go, I think the slide set dropped out. So the pilot phase, there are two sort of steps in the pilot phase. The first step is to bring together existing data, and we established workshops actually since July. So this is all a very recent but very intense effort. We had multiple work groups focus on informing us sort of on what are the key data drivers that we need and must have to sort of make a meaningful effort. And one of the key drivers was this sort of focus that diversity populations across the globe, they have not been utilized. Essentially all effort today in Alzheimer research has been on European ancestry population. So getting phenotypic data that sort of enriches for dementia, focusing on the age range of sort of 50 to 65, maybe up to 70. Go to the next slide, and various cardiovascular phenotypic features and the like. They are sort of the key foundation for the for driving this sort of first phase. And then, we distributed the survey to the sites. As I said, 30 sites engaged, 15 have completed it. And the vast majority of sites have significant number of diversity patients who have responded, that have imaging data, have biomarker data, have GWAS, even exome, and plasma that can be utilized to measure, have done cognitive assessment in a subset of patients, have significant number of patients in the age range of 50 to 65 or 70. So this is a very realistic potential to get that data together for analysis to sort of both drive and inform on this first pilot initiative, that the sites can work together and that we can get diversity cohorts. Polygenic risk score will be utilized in the setting because, that is a tool to sort of predict the disease risk. And if we can add that to ApoE, to family history and alike, we have a substantial sort of, enrichment where we would subsequently, for the second sort of what we call here a higher reach pilot phase, basically select the most informative individuals where the sites have recontact and are able to generate PET scanning, generate imaging data, generate whole genome data, or digital biomarkers or other sort of information. So that is really the focus on the second phase here. So if you go to the next slide, it's sort of shown, I think. Well, I talked about this, so let's just go to the next slide. So this really sort of shows the phase here of the first pilot phase, where we have cohorts. We are going to sort of really use the UK Biobank as the reference site because UK Biobank has a lot of the information of interest, the electronic records, cognitive assessment, certainly not in everybody. And where you see the green, that means that the bulk of the population has that. When you see the orange, it's sort of a significant number of people have it in those cohorts. And when you see red, there's very little data available. And going through DNA, GWAS, exome, imaging, plasma, digital phenotyping, clinical biomarkers, and alike, you can see that there is a significant amount of information available across cohorts from South America, from Middle East, from East Asia, Southeast Asia, Africa, and elsewhere. But there's also significant gaps, and that's what the phase 2 pilot, if you go to the next slide, is sort of aimed at doing. So the first pilot, we estimate that this can be done, this initial data collection for 10,000 individuals for about $2 million. But new data generation sort of on key subjects who have early dementia and would benefit from additional workup. This is obviously a much more expensive phase. And if we were to do all 10,000, it would be in the order of about a $15 million. So this would sort of really be the initial pilot that would be sort of focused on for the next 2 years, and this will set the stage for the ultimate final funding to sort of drive something very unique. That would be ideally diversity cohorts with deep phenotyping to try to resolve the sort of genetic underpinnings and other sort of biological processes of this devastating disease. So if you go to the next slide, this sort of so January is sort of when this will actually be presented to the World Economic Forum now in January at their meeting. That's going to drive the funding for this initial pilot. And the goal is to sort of, get going in Q1 of this year and to reach out to these 30 cohorts, try to get additional cohorts to engage. So we sort of try to get the most informative patients enrolled. And if you go to the next slide, I think that is sort of a closer slide on opportunities for cohort involvement. As Geoff said yesterday, on Monday, I mean, there are now 110 cohorts that have signed up for IHCC. So there is really a major, mass of people who are involved and now engaged. Now we are trying to get everyone to sign a charter. So we have sort of, somewhat of a memorandum or regulatory forum in place for data sharing, and sort of, that we all operate by the rules. We have generated surveys that we've sent out to sites, both with IHCC involvement, DAC involvement, and other initiative involvement. And as we heard from Team A, there is already a significant initiative of bringing IHCC cohort data together in the portal of the atlas that they have. So that is an effort that will continue and now be more focused with for example, the DAC subjects, for that first pilot initiative. Funding of the pilot is this is the responsibility of George Radenberg and his team, and he is very hopeful that this will work and that there will be scaled up funding up to hundreds of millions, potentially sort of from academia industry partnerships going forward. For the publications, for example, the polygenic risk, I think we sort of had a discussion, and the census was that we would like most of the consortium publications just to have a banner and then just describe people's initiative in the acknowledgement that would still be listed on PubMed. Ongoing effort to engage additional cohorts are certainly underway. And this is sort of really by everyone on this call here that we've been trying to get new cohorts to engage. I work quite a bit now with China to get additional data, and Singapore, Africa to get additional data from those countries. And obviously unified effort around COVID is something that, there's a lot of agreement with, and I think we can obviously have significant impact if we put our resources together, and this is also a driver of getting additional cohorts involved with that. And the atlas, of course, is a key tool to sort of, host data to the extent we need data sharing. There are biobanking initiatives. Similarly, if we ever need to centralize biobanking for sort of, future new data generation. So I mean, that's something we will keep in mind. But I think I'll stop here and happy to take questions.
Thanks very much, Hákon. Right on time. Exciting to see a project coming to fruition and talk of the first IHCC scientific publication, which is fantastic, as well as a major new opportunity. As you said, there's some incredible momentum and potentially resource behind the Alzheimer's project. So I think that's an exciting, really exciting new area. And I think, thank you to all the speakers in that session for showcasing some of the science that's been launched. I think we should pay tribute to the people, particularly on the executive committee, who've done some of the hard yards here in terms of forming the collaborations and pulling together the funding to enable some of these pilot projects to get going. We had some discussions on Monday about how IHCC has done a fantastic job of pulling people together and building a framework for science, but now it needs to produce and get some demonstration projects going. And the proof is there that there's some really exciting science going to happen. I think it's also important to point out that there's some projects in specific areas going on there, but those are a tiny fraction of the kinds of things that IHCC cohorts could achieve. And one of the things we need to do as a community is to plan the next stages, which of course is what this meeting's all about, and to make sure that there's real diversity in terms of involving people and cohorts in the planning. So, we are the sum of all of our parts, and if we can really pull together and get more people involved, then that would be excellent. An obvious mechanism to do that would be, as we heard on Monday, about the new committees and the voting that will happen for the new committees. So I'd really encourage people who are enthused by what they've seen here to get involved and to nominate to help shape the process going forwards.
And then in closing, I would just like to thank the wonderful Secretariat team that you saw, whose pictures you saw on Monday, and the great work that they have done to put this meeting together somewhat on short notice, to assemble all of us and to keep us herded as we tend to be cats, to keep us herded in the right direction and to really get us to articulate a lot of things that I think will benefit the IHCC and its members and cohort participants in the future. So Teji is putting thank yous in the chat as well, and I'll just echo the kudos to Eric, Hannah, Meredith, Brittany, Ida, and Laura. So with that, I think you're going to be hearing from us in terms of the schedule that Laura laid out. And if you have anything that you want to talk about anytime about IHCC, please feel free to send notices to me or Terry or Peter or anybody on that advisory group that Laura showed you the pictures of. So thanks again to people on the West Coast as well, that when we started at 7 a.m. or up at 4, and I'm looking at David now, and I see at least there's light. The lights are either on in your house or the sun is shining in through your windows, which it wasn't earlier today. So again, and also colleagues in Asia and in Africa and across the globe who've shared your time with us on Monday and again today. Really excited about this organization and really appreciate everybody's contributions. With that, I'll bid you a good day, good evening, or good morning.